Below you will see the objectives for the innate immunity content for this course. Today's lecture is the first of 5 mini lectures that will cover this content. Today's lecture will focus on the role of complement as a key component of the innate immune system. Now, there are a number of barriers within the innate immune system, including physical barriers such as epithelial barriers and skin, mechanical barriers like mucus and cilia. We're not really going to talk about those much. Instead, we're going to skip straight to talking about chemical barriers such as defensins and complement. Now, defensins, as a brief introduction, are small molecules produced by cells of the innate immune system as well as some epithelial cells. Defensins function to form pores in bacterial cell membranes, and the reason they can do this is they are positively charged peptides that have both hydrophilic and hydrophobic properties. At mucosal surfaces, they're highly enriched in the mucus, so they have a barrier function there. And there are many families of defensins, many family members. Some of these are expressed constitutively, others are induced during inflammatory responses. And I should also mention that defensins are a small molecule that is part of a larger group of small molecule and peptides that have antimicrobial function. But for the most part, in this mini lecture, we're going to focus on the role of complement and the role of complement as a rapid response system to foreign pathogens. And complement refers to a group of more than 30 proteins produced by the liver, secreted into the bloodstream and found in plasma and interstitial fluid. And these are proteases that are inactive, but when they become activated, they lead to a proteolytic cascade that can lead to pathogen destruction. And these complement can become activated by either direct exposure to a pathogen surface or by detecting a pathogen that's coated with antibodies. And there are some similarities here between the complement cascade and the blood coagulation cascade. And importantly, I want you to focus on 3 antimicrobial functions of complement. First, activation of complement leads to direct destruction of pathogens. Second, complement leads to opsonization and phagocytosis of pathogens. And third, complement dramatically enhances inflammatory responses. There are 3 pathways of complement activation: the alternative, lectin, and classical pathways. The classical pathway was the first to be discovered, but the first to be activated is the alternative pathway. It's activated immediately after exposure to a pathogen by exposure to those pathogen membranes. The lectin pathway is the second to act. It has a somewhat delayed response. It's activated when a molecule called mannose-binding lectin binds to the surface of pathogens. And mannose-binding lectin is typically produced within 1 to 2 days after the initiation of an inflammatory response. And last to be activated is the classical pathway. This is induced by antibodies. Antibodies are typically not produced until 5 to 7 days after your primary exposure to a pathogen. However, if you have previously been exposed to a pathogen and are undergoing a secondary exposure, those pre-existing antibodies will, of course, activate complement immediately. So let's focus first on the alternative pathway. And I'm going to start talking about 3 components of complement that are constitutively circulating in your plasma and your interstitial fluid: factor D, C3, and factor B. And factor D is a protease that cleaves C3 and factor B into C3b and BB and C3a and BA. I won't talk about C3a for a few moments, so let's focus first on these 2 components, C3b and BB. C3b, once C3 is cleaved, C3b can covalently bind to the surface of any membrane and associates with BB to form what is called the C3 convertase, or C3b-BB. Now, if this is a mammalian cell, mammalian cells express inhibitory molecules that destabilize this C3 convertase. And so even though C3b might bind to the surface of a mammalian cell, it's rapidly eliminated. But if this is a bacterial cell that doesn't have those inhibitory proteins, this C3 convertase can start to function by cleaving C3 into C3b and C3a. And once that happens, these C3b subunits can start covalently binding and coating the surface of this bacteria. As this reaction proceeds, C3b can also bind to the C3 convertase to form the C5 convertase, which is C3b, BB, C3b. So the functions of the C3 convertase are to cleave C3, release, and C3b can then coat the bacteria. It releases the inflammatory factor C3a, which we'll talk about in a moment, and it leads to the formation of the C5 convertase. Now, the C5 convertase, just as the C3 convertase cleaves C3, the C5 convertase specializes in cleaving C5. And so C5 is cleaved into C5b and C5a. C5b can bind to the surface of the bacteria, whereupon it can now recruit several components of complement, C6, C7, C8, and C9. When these are recruited, together these molecules form a structure that is called the membrane attack complex, or the MAC. And the membrane attack complex essentially functions to form pores in the bacterial membrane. So the C5 convertase cleaves C5 and induces recruitment of C6, 7, 8, and 9, which combine with C5b to form the membrane attack complex and lead to pores in the bacterial membrane and pathogen lysis. And cleavage of C5 also results in the release of this inflammatory factor, C5a. This is an electron micrograph of a bacterial membrane that has been exposed to complement. You can see these little pores now that are coating the surface of this membrane and leading to the lysis of this cell. A second function of complement, however, is opsonization. And so I mentioned that as the C3 convertase cleaves C3 into C3a and C3b, many of these C3b molecules can bind to the surface and coat the surface of this pathogen. That C3b is an opsonin, meaning it enhances the ability of phagocytes to now recognize this pathogen via receptors specific for C3b. This is a process called opsonization. Another common opsonin is IgG. So here's how it works. Here we have a bacteria with C3b coated on its surface. And here we have a phagocyte that has receptors specific for C3b. When these receptors bind to C3b, it allows for phagocytosis and uptake of these bacteria into phagolysosomes within these phagocytes, which eventually leads to destruction of these bacteria. Now, a third function for complement is inflammation. So I've drawn here a capillary with monocytes and neutrophils that are circulating through. These endothelial cells also have receptors for some complement components, including receptors for these cleavage products C5a and C3a. These products bind to these receptors on these endothelial cells and activate these endothelial cells, enhancing the ability of neutrophils and monocytes to migrate into this inflamed tissue. And so this is a key function of C3a and C5a. It activates the endothelial cells, it induces their vasodilation, and increases the migration of these cells into the tissue. It also can act directly on complement receptors expressed by these cells to enhance their activation, and they can act as chemotactic factors as these cells migrate from areas of lower C3a and C5a concentration to areas of higher C3a and C5a concentration, which is where the site of inflammation is. All right, so these are the three pathways. Again, we've talked about the alternative pathway. This is activated by exposure to pathogen membranes. Let's move now to talk about the classical and lectin pathways. So the classical pathway is triggered when antibodies bind to some target on the surface of bacteria. And when this happens, it recruits, and I should mention, this has to be either IgM isotype or IgG isotype. Other isotypes can't do this. So these are the only two isotypes that activate complement. When these antibodies bind, it leads to the recruitment of this molecular complex called C1. And C1 is composed of C1q, C1r, and C1s subunits. C1r and C1s are actually proteases that lead to the cleavage of C2 and C4 into C2b and C4b. When this happens, C4b can bind covalently to the cell surface and associate with C2b and form the classical version of the C3 convertase, C4b and C2b. I do want to mention that there is some confusion about the nomenclature. You will see in some places that this is written as C4b and C2a. It is the same thing. The nomenclature has been updated, and it should read C4b and C2b, but in many places that information has not been updated. So if you see it written either way, it's correct. This C3 convertase then leads to the cleavage of C3 to form C3b and C3a. These C3b molecules can now coat the surface of this bacteria, and C3b can bind to the C3 convertase to now form the C5 convertase, C4b, C2b, C3b. The lectin pathway happens when mannose-binding lectin binds directly to carbohydrate molecules on the surface of bacteria. This mannose-binding lectin is associated with certain proteases called MASP proteases. And these MASP proteases lead to the cleavage of C2a and C4a into C2b and C4b, leading to the formation of a C3 convertase that then cleaves C3 and C3b or C3 into C3b and C3a, coating of the pathogen with C3b and the formation of the C5 convertase. So each of these pathways is triggered by a key protease. In the case of the alternative pathway, it is factor B. In the case of the lectin pathway, it is the MASP proteases. And in the case of the classical pathway, it is the C1 proteases. Each of these pathways leads to the formation of a C3 convertase. The exact makeup of the C3 convertase differs, but each of these C3 convertases specializes in cleaving C3a into C3b and C3a. And once that happens, the pathways are very similar. They converge after the formation of the C3 convertase. Now, I just want to take a moment to discuss regulators of complement. And I mentioned these briefly. There's a number of regulators that are expressed on or associated with mammalian membranes that prevent complement from being activated to destroy your self cells. You would rather the complement, of course, be directed towards foreign pathogens. And so the first one of these is the C1 inhibitor, which, as you might imagine, regulates the classical pathway, particularly the function of C1. Two soluble factors, factor H and factor I, destabilize the C3 convertase, and these factors preferentially associate with mammalian membranes. And there are two membrane-bound factors, membrane cofactor protein, or MCP, and decay accelerating factor, DAF, that also destabilize the C3 convertase. And lastly, there's another membrane-bound factor called CD59, and this destabilizes the membrane attack complex. And again, CD59, DAF, MCP, these are really only expressed on the membranes of mammalian cells. So as you go through today's lecture, or this lecture, you can look at some of these key points to make sure that you have understood all the material that has been covered today.